And that's it.
Great.
Lounging in bed.
Great. Your building is near here, right?
10-minute walk.
Oh, okay.
All right, good. Is it afternoon? It is just afternoon, everybody. Want to welcome you. We have a special presentation today. Our guest speaker is Dr. Anthony Zannas. Dr. Zannas is a psychiatrist. He trained as an MD at the National University of Athens and subsequently went to North Carolina, completed his residency training in psychiatry at Duke, and for the past 3 or 4 years has been a research fellow at the Max Planck Institute, working with Elizabeth Binder. And he's going to talk today about some of the work he's been doing. His CV at this stage of his career is already incredibly impressive in terms of lots of work done understanding epigenetics and aging, and he's just recently written a very interesting review article with George Chroussos that's in Molecular Psychiatry. And we're fortunate to have Dr. Zannas here today to talk with us. The title of his talk is Live Fast, Die Young, the Role of Epigenetics in Stress and Aging.
Great. Thank you. Yeah, hi everyone. I'm Anthony. It's a pleasure to be here, and I want to thank Mary, Neil, and everyone from the chair's office who gave me the opportunity to be here. And it's a I'm very excited to share what I have been working on. So I actually have a slide to give you a little bit of background before I go to the scientific part of the talk. I didn't know there would be an introduction, but broadly I'm interested in how stress can contribute to behavioral and somatic disease phenotypes, so a big umbrella, let's say. But what are the molecular mechanisms that might explain this relationship? And one area I have been very interested in is aging-related disease, so how stress can influence the aging process, can contribute to aging-related disease, and what are the molecular mechanisms that may explain that. So just to give you a very brief background, and Mary mentioned the important points, but so I grew up in the 80s, and in Greece there was this prominent idea that if someone stresses too much, they will age fast, and there still is this idea. It's very embedded in our culture. I guess this was a very big force for me to become interested early on in this relationship. Then I studied medicine in Athens, and after that I spent some time working as a general practitioner in various settings in the military service. The military service is mandatory in Greece, so I had to do that. I worked there as a general practitioner, but also in some Greek islands. And I was really astonished to see many patients presenting, and I had to treat many patients that were presenting with somatic complaints, but they were reporting a lot of stress in their lives when they were presenting with these complaints. So again, this made me even more interested in this relationship. Then right after I finished this clinical work, there was a program, a master's program in stress starting in Greece. And this was led by George Chroussos, so I was very fortunate to do that and learn more about the biology and the science behind what I was interested in. Then I trained as a psychiatrist, and at the time I was very interested in working with elderly population. I found very interesting elderly patients, and I was interested in late life depression and how stress and genes can interact to influence the aging brain. But also at the same time, during my 3rd and 4th year, we had a lot of elective time at Duke, and I took all of that time to do research. And I was very intrigued to, at the time it was, the field of epigenetics was exploding. And it gave me a very nice model about how our environments can be embedded on our genomes and our cells. And this was what attracted me in this field in the first place. Then I wanted to do more molecular work. I'm fascinated by mechanistic work and about signaling pathways. And this is what I have been doing at the Max Planck. This led to an NARSAD award and Marie Curie Fellowship. And then I just finished my PhD a few months ago. So this is just as a general overview to give, just to tell you how I became interested in these questions that I will be talking about now. Okay, so to go to the outline, so again, I will give some background about psychosocial stress and aging-related disease. Then assuming, and please forgive me if this is a wrong assumption, assuming that you don't have a background on epigenetics, I will tell you a little bit about epigenetics and about how stress can influence our epigenomes. And then I will tell you about two projects that I have been working on. One is focusing on a genome-wide biomarker, which is called epigenetic aging. And the other one is focusing on stress-responsive gene that I have been very interested in. So the first is published, the second one is unpublished work. Finally, with some conclusions and future directions. Okay, so as I said, there was this was something that was repeatedly said to me by my grandmother. And my grandfather was saying something a little bit different. He was saying that every disease results from too much stress. So there was, again, this idea that every disease can be linked back to stressful experiences. And then I was interested, as I was growing further, to see characters in novels and movies of individuals that were aging very fast when they were exposed to extreme stressors. At the same time, there were legendary characters that appeared to age very slow when they were leading a very peaceful life. So there was this idea that if we live fast, then we die young. So as I was growing up, okay, my grandparents were right. I was finding evidence, sort of anecdotal evidence, that this might be a true relationship. And just to give you a dramatic example from Victor Hugo's Les Miserables, Hugo says that it was he, Jean Valjean, in fact, the clerk's lamp illuminated his countenance. He was pale and he trembled slightly. His hair, which had still been gray at his arrival to the court, was now entirely white. It had turned white during the hour he had sat there. So you see, this is a dramatic way of depicting that an experience has been very stressful by showing that the individual is aging very fast. And indeed, over the last 1 or 2 decades, there has been a tremendous accumulating evidence showing indeed that different stressful experiences occurring during early life, but also later in life, can contribute to different aging-related disease phenotypes. Of course, the question for me is, what are the molecular mechanisms? And just to give you a hint, beyond epigenetics, of course, which is on the title of the talk, inflammation is a huge link between the two, and that's something that came up in our analysis, I will show you later, and something of my particular interest as well. So obviously, these questions are very important because we now live in an era of what's called the aging crisis. All populations worldwide are aging dramatically. In the next 2 decades, we're expecting to see a rise of 3 times of elderly individuals. Of course, this is a major accomplishment of modern medicine, but at the same time, the problem is that age is itself a major risk factor for all those diseases that are the leading causes of morbidity and mortality, and we really have to advance our understanding about how we can prevent these diseases. And of course, stress is ubiquitous and potentially preventable, and potentially we can identify molecular mechanisms that we can then target to prevent these diseases. Okay, so now to give you a sort of a primer on stress-induced epigenesis, the way I like to call it. So actually the basic ideas, the foundation of what we call epigenetics was laid very early on by Aristotle in the 4th century before Christ. He was actually studying very crudely chicken embryos, and he saw that the embryos were not absolutely preformed, so they were taking different directions depending on the environmental exposures. This was somehow forgotten in a sense, and then there was a major figure that came about, Baptiste Lamarck, and the very important idea that he brought was the idea of the soft inheritance. So there is a trait that can be shaped by environmental exposure, and this can actually be transmitted to subsequent generations. And now we have more and more evidence that this can actually happen also with stressful experiences. Another major figure was Conrad Waddington a little bit later, and he brought up the idea of the epigenotype. So there is some stable trait that is actually not traced to the genetic code itself. And then in the 70s and 80s, we actually discovered that there is a functional DNA methylation. So DNA methylation can have functional consequences. And now we are in a very exciting era because we are in an era where we can sample quite easily, still somewhat expensive, but very easily from a methodological perspective, many sites across our epigenomes, and actually we can even do methylome-wide sequencing. And this is what has brought a revolution in the human world. So the way we use epigenetics nowadays is the constellation of all those mechanisms that can influence gene expression and phenotypic expression without changing the genetic code itself. And there are several mechanisms, as you can see. There's DNA methylation, there are histone post-translational modifications, there are RNA-based mechanisms, there are more and more, so the repertoire keeps increasing. And I will sort of focus, without ignoring the other mechanisms. Everything is very important, and we have to integrate all these mechanisms. But I will focus on DNA methylation, again, because thanks to DNA methylation arrays, that's what we can study most in humans. Okay, again, I was very interested in epigenetics in the first place because it gives a very nice model, sort of a molecular interface between the environmental exposures, and our genomes. So it's really intriguing, but we don't know how initially these dynamic epigenetic marks are stabilized over time, perhaps through repetitive exposures, and finally contribute to these phenotypes over the long term. But then the question is, how can stress among these environmental exposures can, let's say, get under the skin and impact our epigenomes? This is something that I have been very interested in. And for that, I have been focusing on the HPA axis, hypothalamic pituitary adrenal axis. And this is by no means the only effector, the only mediator that is important. But it is a very nice one that we can use as an example, because we know quite a bit about it. So what we know is that when we are stressed, when there is a stressful experience detected in the brain, we have secretion of corticotropin-releasing hormone by the hypothalamus. Then we have secretion of ACTH by the hypophysis, and all this culminates in peripheral secretion of cortisol. And then the way cortisol works, and this is not the only way it works, but it is a major way that it does. Cortisol traverses essentially all our cells and binds to the glucocorticoid receptor, which is a transcription factor, a ligand-dependent transcription factor, that when activated, translocates inside the nucleus and binds to these conserved elements in our genome, so some sequences that are there to be detected by the glucocorticoid receptor. And beyond acutely changing transcription, what has been shown, intriguingly, is that by the binding of the GR to these sites can also induce lasting epigenetic changes. So it's a sort of a molecular memory. So the cell remembers that it was exposed to glucocorticoids, and this will influence how the cell will respond to subsequent glucocorticoid exposure, or if you prefer, stress exposure. And just to zoom in and give you a little bit of more about this mechanism. So as I said, when we are stressed, we may have deregulation of glucocorticoid signaling. We may have aberrant cortisol levels. In turn, this can lead to aberrant activation of the glucocorticoid receptor. And then this will influence how much activated GR we have inside the nucleus. And this can bring about a series of events. On one side, we may have binding of the GR through other transcription factors, so through interaction with other transcription factors, binding to the genome and changes in methylation levels, which have been shown to be particularly at gene promoters. Or, and this is one mechanism that we have studied a little bit more than the other one, we may have direct binding of the GR to glucocorticoid response elements, and this can lead to demethylation happening, as I showed you also in the previous slide, demethylation happening at or near these glucocorticoid response elements. Finally, of course, we can have a series of other mechanisms, and I'm not showing everything here. histone modifications, again through binding through other transcription factors or through direct binding to GREs. So, in fact, it has been shown that stressful experience occurring throughout our lifetime at different time points can lead to lasting epigenetic changes like the ones I showed you. So stress occurring as early as in utero, later in childhood, adolescence, and so on, can lead to this remodeling of our epigenomes. And what we have proposed recently, yeah, sure. GREs are not cell type specific because they are the genetic based on the genetic code. But however, so they are present in principle in all our cells, but they are not functional in ours. So the accessibility to GREs can be cell specific. But in general, GREs tend to be cell tissue independent. But the other mechanisms I showed you of interaction of the GR to the genome are more tissue specific. So in general, there is some tissue specificity, just to shortly answer your question. But
GREs are mapped across the different cell types using some genomic
yes,
approaches.
yes.
They're pretty well known, or there's something that's predicted? Or?
It is pretty well known. So if you look at the ENCODE data, they have ChIP-seq data, and you can see that many GREs are not functional in all cells. So depending on its cell type, they will have different accessibility.
Oh, so they are cell type specific then?
Yes, but not completely. So there are many common GREs. So yeah, I don't know exactly the percentage of how many. The other mechanism that I showed you earlier, yeah, this side of things is more tissue specific. The GREs tend to be more common across tissues, but again, there is some tissue specificity. Hope I answered the question. Yeah. Okay. Yeah. So what we proposed is, let's imagine that we have, and I mean there is some evidence to that. It's not something entirely new, but we said maybe there is a cumulative. There is an accumulation of these epigenetic events. So, and when I say accumulation, I don't mean only, it can be quantitative at specific sites. So these specific sites that are stress sensitive will accumulate over time through repetitive stress exposure, these epigenetic changes. So we will have a quantitative, a dose-dependent effect if you prefer. But also, we can think of it at a systems level. So it's as these sites are influenced, so different sites are influenced across, that are scattered across our genomes. In the end, we may cross a threshold that we can see at a systems level a biological effect. And I think it's a very nice model to think about that. And very relevant to bring it back to aging. I think it's a very relevant idea because we know that epigenetic alterations is one of the hallmarks of the aging process. So actually some people propose that epigenetic regulation may be causal for the aging process. And this, of course, has not been proven yet. Okay, so now to go to show you some data. As I mentioned, first I will show you a project focusing on a genome-wide marker. I have been very interested in what's called epigenetic clock. And the way these markers work is they are composite predictors combining the status of multiple methylation sites. Again, these sites are interrogated through DNA methylation arrays. And so they take all these sites. Each of these sites is weighted. So you have a beta coefficient for each site in the regression model. And then, in the end, this model gives you an estimate of the individual's age. So this is what we call DNA methylation age. Intriguingly, these predictors can give you a very accurate prediction of how old is someone, more than 90%. So just by knowing the methylation status of our genomes, we can predict very precisely how old we are. So the idea behind that is that if we had a perfect correlation between chronological age, and this is the same idea like every measure of biological aging. If we have a perfect correlation between the two, we would have this line between how well we can predict chronological age with our biological predictors. However, we know that there are certain individuals that appear to evade biological aging. And one famous example is Sean Connery, who within these 20 years when these pictures were taken, he showed basically no signs of additional biological aging. On the other hand, there are individuals that appear to age very fast, and one example is Barack Obama. You see, during the 8 years that he was a president, he showed clear signs of biological aging. So, of course, for me, the question is how can life adversity change the ticking rate of our biological clocks and epigenetic clocks? The idea is that if we subtract chronological age from DNA methylation age, we have what's called epigenetic age acceleration, and this is a marker that has been used in the literature, or delta age. I will call it delta age. And this delta age, or epigenetic age acceleration, has been shown to predict a large number of already aging-related disease phenotypes. There is an explosion of studies examining this marker with different phenotypes. You can see all-cause mortality, cancers, cardiovascular disease, and so on. So it does appear to hold some promise as a biomarker for aging-related disease. To give you, so we wanted, of course, to test if stress can influence this marker, but first I will show you some how well you can predict, just to, because for me, the first time I saw that it was incredible. You see that you have this nice line of prediction between DNA methylation age and chronological age. Now we have tested this in many more cohorts. I'm showing you 2 cohorts where we did that. You see correlation coefficients that exceed 90%. But as I mentioned earlier, despite this very nice line of correlation, you see that there are individuals that fall either below or above this line. And the question is, how well can we explain this variance by stressful life experiences? And this is what we wanted to test. Yes?
For cells, this assay was done on
Yeah. I didn't mention, that's good. So this was in blood, whole blood. Yeah. I mean, yeah, its point is one individual, and this was just to also give you a little bit of background. So this is the Grady Trauma Project, and these are individuals that are highly traumatized, so have been exposed to a lot of stress. And this is an in-house cohort. We have the Max Planck cohort where we have individuals that are depressed or control individuals. Okay, so we tested this in the Grady where we have, as I mentioned, the individuals are highly stressed. We have these stress questionnaires. So we have documented exposure to stressful life experiences. You see, there was a positive correlation between cumulative lifetime stress. So that's what the questionnaire answers. And delta age. So the positive correlation meaning that the more stress you have been exposed to, the more you have accelerated by epigenetic aging. But what was also interesting was if we stratified individuals in older and younger, we could only see this relationship in older individuals. And this was after controlling for the number of life events, because of course older individuals may have accumulated more stressful experiences. So this suggested that maybe there is an interaction between the aging process itself and stressful experiences that, or maybe a wear and tear effect that could explain this relationship as individuals grow older. Another interesting sort of secondary analysis was when we looked at personal stress, so stress that affects the individual directly, versus network stress, so stressors that affect the individual's network. And we saw that only personal stress was positively associated with epigenetic aging. And the last thing, as we did as a secondary analysis at this level, and was somewhat surprising to me, was that when I looked at individuals that were exposed to more or less childhood stress, so individuals that have been exposed to more childhood stress is the red line. So those individuals were actually somehow protected to total stress exposure throughout their lifetime. So there appeared to be a resilient phenomenon. This actually fits with some animal work showing that what's called the match-mismatch hypothesis. So mice that have been exposed, for example, to early life stress are sort of resilient, as long as this stress has not exceeded certain threshold, are resilient to subsequent stress exposure. Okay, so of course, yeah, just one more thing. So there was no relationship between epigenetic aging and childhood stress or recent stress. So again, it was this idea that it is the cumulative stress, the stress that accumulates throughout our lives that is associated with epigenetic aging. And for me, of course, the next very intriguing question was what can be a mechanism behind that? And of course, as I mentioned, I'm interested in HPA axis. And could it be that glucocorticoid signaling gets deregulated and is driving these epigenetic changes? And to start with, we wanted to see if these sites may be more sensitive than average methylation sites to glucocorticoids. And for that, we did an enrichment analysis where we looked at annotated data, so online available data, and we first asked the question, how many of these sites fall right within a glucocorticoid response element? And this is the red dotted line. So the number was 85. And then we said, let's sample random sites throughout the DNA methylation array and see how many we would expect by chance. How many sites we would expect by chance. And this is the random distribution. So you see this distribution if you just sample randomly throughout the epigenome. And as you can see, the red dotted line is much higher. The number is much higher than what we would expect by chance, significantly higher, which then supports the hypothesis that these sites are more sensitive than average sites to glucocorticoid exposure, or this is something that we would expect. And we actually tested this because we have data of individuals that have been exposed to glucocorticoids, and we have DNA methylation before and after exposure. And we saw indeed that 1/3 of these sites is sensitive to glucocorticoids. They either increase or decrease, but as you see, most of them get demethylated. And as I mentioned again, this goes along with this mechanism because we know that when glucocorticoids bind to glucocorticoid response elements, we usually have a demethylation. So this agrees with the mechanism. But another thing I wanted to point you here is that, on the x-axis we have distance to glucocorticoid response elements. And what each of these dots is one methylation site. And if the dot exceeds the red dotted line, it is statistically significant. So as you can see, the closer we get to the glucocorticoid response element, and 0 is right within the glucocorticoid response element, the more likely are these sites to exceed the level of statistical significance, so to respond to glucocorticoids. So this altogether really was in line with the mechanism, the potential mechanism that glucocorticoid signaling can be driving these effects. And just one, so then we did sort of functional analysis. We wanted to see if we go to genes with transcription start sites close to these epigenetic clock sites, how many of them will respond to glucocorticoids. And the number was very high. It was more than 80%. Usually, we see changes in transcription levels of about 20% of the genes upon exposure to glucocorticoids. So this was a very high number. And several of these genes were immune-related genes, DNA damage response genes, and so on. And we did an enrichment analysis which showed an association with leukemia, arteriosclerosis, and coronary artery disease, suggesting that these diseases can be influenced by stress through epigenetic effects. So just to summarize this part, we see that cumulative lifetime stress, but not recent or child stress alone, accelerates epigenetic aging. The effects appear to be evident only in older ages, and the question is it a wear and tear effect of the epigenome? There is a component of programmed aging that is playing into that? And then glucocorticoid signaling, we think, may mediate the effects of life stress on the epigenome along the lifespan, and we are doing some cell culture work trying to really pinpoint what exactly is the mechanism behind that. Okay, so, yes?
Older ages, what age group are you talking about?
The age range was between 18 and 75.
No, the older age group.
The older age group. So the older, I just did the median split of the subject. So everyone that was above 44 was older, and everything that was below 44 was young. So it's sort of a crude, but I wanted to do this stratification to see.
Along
Yeah.
that line,
Mhm.
question earlier. How can we combine the cohort younger and older and more significant in terms of the correlation between the delta age?
You are referring to the p-values, right, that I showed? Yeah, the reason is just the sample size. The effect size was higher in older, but I think the p-value is lower because of the sample size. So the thing is,
But you should also have an averaging effect because in the younger you don't have much effect. Once you combine the two, you actually should be more of an average.
yeah. So the effect size is average. If you, I mean, yeah, it's sort of hard to see, and I don't have the effect size, the beta coefficients here. It is average, the effect size, but the p-value is lower because you see there are much less individuals here just because of the stratification.
So much bigger beta.
Yeah, so the beta is much bigger here. Yeah. I think it was double or close to double. I don't remember exactly. Yes?
So I have a couple of questions about the clock idea.
Mhm.
One possibility when you take blood, there's probably different kinds of cells in the blood that you're looking at. And so one possibility is the ratio of those cells is shifting as a function of age, and that's the readout that you're getting. Or the other possibility is that really these markers in all or some subset of cells are changing as a function of age. Do you have any thoughts or data?
That's a great question. Yeah, so for everything I showed you here, we are controlling the blood cell composition. It's not the perfect way to do it, but it is a very precise from what others have shown. So the way we do it is with DNA methylation array data. There are certain sites, certain methylation sites, with which you can predict the blood cell composition. So consistently, these sites are differentially methylated across different subtypes. And then we do this we put this in the regression model as covariates.
Okay.
This is sort of a standard way to do it. If you don't do it, you cannot publish this study.
Okay, good. And then the other thing is it seems like you're getting evidence that the glucocorticoids are changing this clock signature, kind of changing the clock time. But is that a transient effect, or is that really a durable effect, right? People all the time have spikes either because they have a stressful period that's acute or they get glucocorticoids as a drug.
Mm-hmm. The limitation of the data I showed you is that they're cross-sectional. So to really answer this question, we would need longitudinal data, which I would love to have in the future. We did DNA. We checked epigenetic clock before and after glucocorticoids, and it didn't change within this short time frame.
Good.
So I mean, all the data point that we need the chronic stress to see a durable effect. But that's as far as I can tell for now.
Thanks.
Okay, so now I will take you through another, and actually it's a project that I find very exciting because I was able to combine a lot of different expertise and dig more into the mechanism of things. So for this, we are interested in stress-responsive genes, in particular stress-responsive genes. And one gene that we have been working on in our institute is FKB5. The reason we're interested in this gene is that it is one of those molecules that it encodes for one of those molecules that can really influence glucocorticoid signaling. So far I told you that when the GR is activated, it goes inside the nucleus, but it is not an all or none phenomenon. So we can have different rates of translocation inside the nucleus. And when FKB5 is bound to the glucocorticoid receptor complex, it can slow down the GR. So it has a sort of an inhibitory effect. But when, for one reason or another, the GR is activated, it goes inside the nucleus, and among the genes that respond to the glucocorticoid receptor is FKB5 itself. Now, the interesting thing in that is that we have an induction of FKB5, and this in turn creates this ultra-short negative feedback loop that can regulate glucocorticoid signaling. And as you can see, the reason that the FKB5 responds to GR is that we have all these GREs scattered across the gene. And actually, FKB5 is the most glucocorticoid responsive gene in several tissues, including blood. And this is, as I mentioned, we are studying blood. So this was a very relevant gene to examine in this question. And actually, when I started working on this, there was already something known about aging in the FKB5. So there were some data in post-mortem human brain, but not in blood, showing that FKB5 can be upregulated with increasing age. And actually, I'm not showing it here, but also in individuals with Alzheimer's disease. And there were some data showing that the FKB5 gets demethylated again in post-mortem brain with increasing age. So I was very interested to see if we have the same phenomenon in peripheral blood and what that might mean. So here I'm showing you, I hope you can see the colors, but it is 3 different cohorts. Again, this is the Grady Trauma project. This is a huge epidemiological study in Germany, the CORA. And this is the Max Planck Institute study. So it's in total close to 3,000 individuals. And you see that there is this consistent pattern of demethylation. I'm sorry, can't point. Demethylation with increasing age. And to do that, I took all the sites across the FKB5 that were covered from the RA. I checked all of them, and it was always the same 2 sites. Actually, I have now tested that in 6 cohorts. It's always the same 2 sites that are within the FKB5 promoter that selectively are influenced with increasing age. But as you can see here, I hope you can see, is that although we have this nice slope with increasing age, well, I don't know if it's nice, but we do see a moderating effect with depression. So individuals that are depressed appear to have an accelerating course of decrease in methylation with increasing age, which makes sense because individuals that are depressed have deregulation of stress responses, and this is a stress-responsive gene. So somehow it gets demethylated more in those individuals. So these are, and just to mention briefly, these are 2 sites, which here I provide the average of the 2 sites because they're very close to each other. They correlate, so just to simplify things, we average these 2 sites. And this is from another cohort. It's the so-called Helsinki birth cohort study. These individuals were separated when they were very young. They were sent away. They were Finnish kids that were sent away from their parents to be protected from the Nazis during Second World War. And again, we saw that with these extreme stressors, they were separated for 1 to 2 years from their parents. We again saw a decrease in methylation that was very interestingly selective in these 2 sites. We would not see it in other FKB5 sites. So there really seemed to be a stress effect that was selective for these age-regulated sites. Of course, I wanted to understand what is the functional consequence of that. So then I correlated age-regulated sites with FKB5 expression. You see there is this modest negative correlation between the 2, something we would expect since the 2 sites are located within the gene promoter. But what was more interesting is when we took into equation cortisol. So as I mentioned, FKB5 is a stress-responsive, cortisol-responsive gene. So we would expect a positive relationship between cortisol and FKB5. But when we stratified individuals in low and high methylation levels, we saw that individuals that had demethylated locus at those age-regulated sites, they had a much stronger relationship between cortisol and FKB5. So the hypothesis behind that was that the locus gets de-repressed, and then we have more high response to the same levels of cortisol that could explain this relationship. And the same was true for when we stratified individuals in older and younger. So again, individuals that were older seem to have a stronger relationship between cortisol and FKB5, which could be explained by, at least in part, by this age-related demethylation. Okay, and this is, I will show you some data that I just finished, some experiments I finished last week. I wanted to understand at the cellular level to understand the mechanism behind this relationship that I showed you. So how can the gene get de-repressed? Can we really prove that using cell lines? And here I'm showing you the 2 sites. These are the 2 CTGs, the 2 methylation sites that are age-regulated. And I was expecting to find either between these sites or close to these sites a glucocorticoid response element that could explain this relationship. But interestingly, instead of finding a GRE, I found a kappa B response element. So it's an element that where NF-kappa B can bind. And the closest glucocorticoid response element, which is actually not a glucocorticoid response element, but it's an androgen response element, this is from our discussion. This is a little bit more 500 base pairs upstream. We have this androgen response element where also glucocorticoids can bind. And then a little bit downstream, as I mentioned, this is in the gene promoter. So a little bit downstream, we have the transcription start site. So then what could happen here? So we hypothesized that we don't have direct binding of the GR, but we have tethering of the GR through binding to NF-kappa B. And then when we have demethylation of these sites, perhaps we have derepression of the gene, supposing that also NF-kappa B is around to allow the GR to bind to this site. That was the hypothesis. And I designed 2 experiments to test that. The first was a reverse chip experiment, so-called reverse chip, where we took biotinylated probes with this site, and we planned to in vitro methylate, and then see if we have differential binding of GR and NF-kappa B using antibodies to this site. And the second experiment was a reporter gene assay, where we used a CpG-free luciferase reporter vector, and you want to use this vector so that you don't have other methylation sites that can interfere with your in vitro methylation assay. And this is what we got from the reporter assay. This is what I finished last week. So it's very interesting. So when we in vitro methylate, we have, you see, at baseline, we have a substantial decrease of the luciferase signal, so about 40%. So there does seem to be an effect at baseline, but what is also very interesting is when we treat, this is dexamethasone again, so a glucocorticoid receptor agonist, you see you have some induction in the unmethylated state, but you have no induction in the methylated state. So this is taken away when the locus is methylated. And this is, we know for sure that these methylation sites are driving this effect because that's the stretch of the gene that I took was only those 2 sites. And when we stimulate also with NF-kappa B, we have an additive effect. When both DEX and NF-kappa B are around, we have an additive effect. And again, we have almost no induction when the locus is methylated. So really, this supports all the human data that I showed you. And unfortunately, I don't have the chip data yet. I hope next time to give you an update. Yeah, I mean, we have to prove it also at the chip level to really show that there is differential binding of GR and NF-kappa B to this site. Okay. And then the question for me was, what can this bring about? Okay, we have a deregulation or differential regulation of FKBP5. What does that mean for the cell? What does that mean for the organism? And for that, I used genome-wide gene expression data. And I tried to find all those genes that are co-regulated with FKB5. So I just did correlations between FKB5 and all the other genes. All the genes that correlate with FKB5 are what I call here FKB5-related genes. As you can see, it's a very large number, but this is something we sort of expected because this is a very versatile protein and it interacts with a lot of signaling pathways. So biologically, this could make sense. And then I did a disease association analysis which pointed to inflammation as a very relevant process, and transcription factor target analysis which intriguingly pointed again to NF-kappa B. So it appeared that when we have deregulation of FKB5, we have an inflammatory process, and this is all human data, genome-wide gene expression, blood data. So we have an inflammatory process that is driven by NF-kappa B. And then again, we went to cell lines using Urca T cell line as a model system. And we wanted to really prove that when we have upregulation of FKB5, we have heightened inflammation and more NF-kappa B signaling. And this is just a control experiment where we see we overexpress, we transfect cells with FKB5 construct. And we can prove that we have more FKB5. And then I did an NF-kappa B reporter activity, a luciferase assay. Again, when we overexpress, this supports the data, so we have more NF-kappa B signaling. And here we tested interleukin-8 because it is a very important chemotactic factor. And sort of I skipped a little bit of the data, but FKB5 also correlated in our data with more neutrophils in periphery. So we hypothesized that we have heightened chemotaxis when we have more FKB5 that is driving this inflammatory effect. So again, the cell line, the cellular models here supported the human data that I showed you, that we have this inflammatory effect that is driven by NF-kappa B. But then we also wanted to understand who is the important player in NF-kappa B. How can FKB5 influence NF-kappa B signaling? And we wanted to dissect the NF-kappa B pathway. And here I show you again gene expression data where we have taken all the NF-kappa B network, and each edge in this scheme is a pairwise correlation between partners that are part of the NF-kappa B pathway. And these pairwise correlations we then control for the expression of all other partners in the pathway. So from the transition to lower, between lower and higher FKB5 expression levels, you can see that many go from, for example, from a dotted line, which is a negative correlation, to a continuous line, which is a positive correlation. So there are substantial changes between the two networks. But the most important one is the one I have highlighted here in orange, if you can see. Of course, the names don't mean anything unless you look more carefully at the NF-kappa B pathway. And it is intriguing because these two genes encode two very important kinases, which are part of the alternative pathway, alternative NF-kappa pathway, or the non-canonical NF-kappa pathway. And these two kinases decide how much it will be activated, the alternative pathway. So the way this works is that one of the two kinases, NIC, phosphorylates IKK alpha. When IKK alpha is phosphorylated, it is more active, and then we have more NF-kappa B signaling. So the natural question, as I mentioned, FKB5 is a chaperone and a very versatile protein that interacts with many different players. The natural question was, if FKB5 now interacts at the protein level, forms a complex with NIC and IKK alpha, and this can regulate NF-kappa B signaling differentially when we have different levels of FKB5. And this could explain the changes that we see at the gene expression level. And we tested that, again, in cell models. We're using Urkat T cell lines and peripheral blood monocytes from healthy individuals. And indeed, there was an interaction between FKB5, NIC, and IKK alpha. So the three of them appear to form a complex, a common protein complex. But what was very interesting is that when we treat with dexamethasone, we have an increase. As you can see, these are just co-immunoprecipitation experiments pulling from different sides to really prove that the complex is coming together. And we could see that all these interactions were going up when we were treating the cells with dexamethasone. So the complex was really becoming stronger. But also very interestingly, when we were treating with FKB5 antagonists, and this is a drug that we have in our institute, we could prevent this strengthening from happening. So co-treating cells with FKB5 antagonists and dexamethasone could prevent the glucocorticoid-induced increase in this complex, in strengthening of this complex. So this sort of proved that this effect of glucocorticoids was happening through FKB5. I hope this is clear. If you have any questions, please interrupt me.
What does the drug do exactly to FKBP5?
Yeah, so it's interfering with protein interaction. So it's FKB5 is a chaperone and interacts with different. It changes the conformation of the.
So it can't bind the glucocorticoid receptor, you mean, or what?
It binds the glucocorticoid. It binds, but with less affinity.
Okay.
With the GR. Yeah. Okay. Yeah, and then we did, as I mentioned, when IKK alpha is phosphorylated, is more active. So we saw again the same pattern. So when we treat with dexamethasone, we have increased phospho-IKK alpha, and this is corrected over total IKK alpha. And with this, we can prevent again with FKP5 antagonists. And this is again the same is when we ectopically overexpress FKP5, more phospho-IKK alpha, we can bring it back to FKP5 antagonists. And the same is true for NF-kappa B luciferase reporter activity. Again, when we overexpress FKP5, we have more NF-kappa B, we can bring it back to baseline with FKP5 antagonists. And this is just to summarize. So as I mentioned, we have a complex between FKP5, IKK alpha, and NIC. And this, at baseline, we hypothesize that it has relatively lower activity. When we treat with DEX, this upregulates FKP5, or maybe we can overexpress FKP5, and this complex comes together much stronger. Now we have phosphorylation of IKK alpha, and this induces NF-kappa B activity. And all of that we can prevent with treating with FKP5 antagonists. And then because inflammation is a major risk factor for myocardial infarction and for acute coronary events, we looked back and said maybe we, in individuals that have history of myocardial infarction, we would see a difference in methylation levels. And indeed, you can see from two different cohorts, we had in individuals with a positive history of myocardial infarction, we found lower methylation of these age-regulated sites. With the limitation here, of course, being you see, we have only very few individuals that do report a history of myocardial infarction because our data sets are not aging-related disease-focused, they are more stress phenotype-focused.
Are they age-matched, those two groups?
No. They are not.
Are the MI ones just older? I mean, wouldn't that?
Yeah, they're only. I only controlled in the regression model for AIDS.
Okay.
But they are not age matched.
So what we're seeing there is not adjusted for age.
It is adjusted.
It is adjusted.
So in the figure it is not.
Result is adjusted,
Exactly. Yep. Okay, so just to give you a schematic summary, hope I made it clear, but. So we have these two methylation sites that are upstream from the FKP5 transcription site. We have a combined effect of aging and childhood trauma and major depression. We have demethylation of these sites. This leads to a higher induction of FKP5 and higher NF-kappa B activity. We can change the trajectory by treating with FKP5 antagonists. Intriguingly, as I mentioned earlier, we have this kappa B response element right upstream from the transcription start site. So there appears to be a positive feedback loop. So when we have more NF-kappa B, we again have more FKP5, and then this brings the whole system to a new level, which unfortunately, of course, downstream leads to more inflammation, more chemotaxis, and we think also high risk for myocardial infarction. Okay. So to conclude everything, and I think I'm running out of time, and to give some future direction. So I'm very interested in the molecular mechanisms that link stress-related phenotypes with disease risk. And I think there are tremendous possibilities to examine this relationship with the tools we have today. And I'm very interested in using this model of multi-omic approaches in humans and then dissecting the mechanism. As I showed you, using cellular models, I'm also very interested in using induced pluripotent stem cells as a model. And of course, this can go both ways. Someone can start from the mechanism and then go back to human cohorts. And I have been very fortunate to be in an environment where, there was a very nice crosstalk between scientists from all these different disciplines. And of course, the data I showed you is the result of that. And some key questions is, for example, what is the timing, the duration, the intensity, the type of the stressor that can lead, and some questions that came up during this session. So how can this be stabilized? How does it work? So of course, these are questions that we can only answer with longitudinal designs, much more to get more deeper understanding on how this works. I'm very interested in identifying novel molecular effectors and mediators of stress. And again, using omic approaches, I think there are tremendous possibilities to do that. Of course, a big question is whether we have the potential to identify stress-induced molecular changes as biomarkers. And of course, how much of that is vulnerability biomarker, how much is resilience, or how much is just epiphenomena. So this is something that, I mean, I showed you some markers that are changing, but what does it really mean? Does it really lead to higher disease risk? How does that work? And then, of course, in the longer term, whether we can use some of this data to identify novel treatment candidates, whether we can prevent and reverse some of these with certain interventions. So I talked a little bit about cellular models and how I like using that to dissect the mechanism, but there is also a very, very interesting crosstalk with animal work with, for example, with rodent models where we have very nice stress models, I feel. And one example is, I mean, someone can use potentially translatable biomarkers. And just to give you an example using the epigenetic clock, these are again our data in humans. But very recently, there was an epigenetic clock developed also in mice. Actually, there are now 2 epigenetic clocks developed in mice. And you can see the same pattern. You can see a very nice correlation between predicted and chronological age of the mouse. So I think there is potential to use such biomarkers with obvious limitations. For example, in this case, the sites are not the same. But when we take the methylation sites from humans, we can still predict the chronological age of the mouse. So in the end, the idea would be to elucidate conserved mediators, pathways, and pathways of stress-related disease. And here are just some examples in work in humans showing that the epigenetic clock, the epigenetic aging can be, the effect of stress on epigenetic aging can be prevented when we target, in this case, children that are high risk for this effect to happen. And there is a big literature in mice in rodents showing that we can prevent stress-induced epigenetic changes by treating with epigenetic drugs. Okay, so in general, I mean, when someone thinks of anti-stress aging interventions, they think of something very fancy, but I think it can be something very simple, going to your favorite island or coming to San Diego and going to a beach, and there maybe we can relax our epigenomes. So, yeah, just to mention, Elizabeth has been a great mentor, and she allowed me to develop this niche that I showed you. And of course, many people helped with the data. As I mentioned, we have a very nice interdisciplinary environment at Max Planck. And yeah, so I'm happy to take any questions. I think I'm right on time, but yeah. It's really a pleasure to be here. Hi. Yes.
Anthony, this is really very nice combination of looking at genomic events and cell signaling. With the pathways you're showing with the MAP kinase pathways, do you have any idea what proteins might be forming the scaffold? There's a lot of data. I mean, in the GPCR field, there's a lot of data like beta-arrestins primarily hit GPCRs, but they also hit steroid receptors. It's a very interesting story about androgen receptors. A collaborator of mine has shown this for the glucocorticoid receptor in terms of cardiac phenotypes. But I just wonder how those scaffolds get formed and then traffic the receptor into an interesting pathway that is involved in aging, like you're suggesting.
Yeah. Well, we know that the trafficking of the GR is through a dynein mechanism. Is that the question? The question is about FKB5 and GR, or?
He was suggesting how these scaffolds got made between
NF-kappa B.
NF-kappa B, the FKBP5, and there's some glucocorticoid interaction, and then it gets trafficked into the MAP kinase pathway and the CHUK pathway. The two kinase pathways you showed.
I don't know. Well, we don't know how the scaffolding is happening. I mean, we know that these are both kinases. It must be transient chains. But yeah, I don't know how the scaffolding. Any ideas?
Well, things phosphorylate. I mean, you have a number of phosphorylation events, so there has to be. I don't know if it's tyrosines or series 3 means
Mm.
affinity change. I think you're showing these interesting affinitive changes where these things come together
Yeah.
and form this complex that then moves the receptor into the cytosol for some signaling pattern. Because there's targets there that can be looked at. I know you have some targets that you're looking at for FKBP5.
Yeah.
It's a connection that I think is very nice the way you made it. You don't see that often.
So yeah, I would be very interested to understand more how it works then.
About how many predictors are used in the model, the aging clock? What are we calling it?
353, the methylation sites.
Okay.
Yeah.
And it's the same model. It's the one proposed by Horvath, and then everybody's
Yeah,
using
exactly.
exactly that model.
We
And-
use the Horvath predictor.
Okay. And some of them are methylation sites, but then you also mentioned that there was some way of dealing with the cell type. Is that also using methylation sites, do you mean, or that's using some other kind of data?
For the cell composition, we use methylation data
Okay.
again, but
So
it's
that's some of those 353 are
no, these are different
a
sites.
separate set. Okay.
Yeah.
And how many is that? A small number or a large number?
I don't remember the number. Maybe
Similar number, maybe? or-
200. Yeah, I don't remember the
Okay.
exact number. I think it's in the hundreds.
Right. Okay. So it's a small number.
Mm-hmm.
And they have approximately equal weights? Like you mentioned that each one of them is given a weight. So of those 353, are some of them accounting for most of the signal, or is it really necessary to have that many?
So they have weights based on how well they correlate with chronological age.
Right.
And
Do those weights have a wide range or not?
I don't
A
remember
very specific question. I was looking at the paper, but I'm not immediately able to figure it out. So
I can send you. I have a table of
Yeah.
so there is a table with the beta coefficients,
yeah, right.
and but
Models other than Horvath, right? That they're
Yeah.
similar.
it's the one we used is the one developed by Steve Horvath.
Yeah, but there are also some others.
Yes.
But I don't think they differ that much.
Mhm.
To which
Performance or what they use.
or which sites they use.
They actually differ. They differ in the sites they use, which is interesting. Yeah.
Okay.
There are different sites. So there is one with 70 sites. There is one actually with only three sites. But the Horvath outperforms all of them. And it also outperforms telomeres as a biomarker value.
Yeah, I have a question that's actually not in my area of expertise, but we, in our HIV neuro AIDS program, we do have a couple of investigators who've been interested in this FK, 5, and but I guess there's a 51 and a 52, and that one is more pro-inflammatory and one is more settling down the system. So is the one you're talking about what's called the 51 here?
Yes, exactly. Just to show you.
And what about the other one?
Yeah, I have it on the slide. Okay.
Maybe I missed it. Yeah.
So no, I didn't mention. So this is FKB51. So there is some sort of discrepancy in the literature. Some call the protein FKB51 and the gene FKB5. I like to call FKB5 both.
Okay.
It's just different camps. And this is the FKBP52 or FKBP4. And they are thought, at least in glucocorticoid receptor signal, to have opposite effects. As you mentioned, one, this is promoting GR signaling, and the other one, FKB5, is inhibiting GR signaling.
I just didn't know if you were looking at both of them.
Yeah, for this one, I looked only at FKB5, but in general, we're also interested in FKB4, and we're doing some assays. With aging, I checked this one, and it doesn't change with aging, so that's why I focused on. Yeah, but this is a good question.
Good, thanks. As I say, this is not exactly my area, but I know
Mm-hmm.
some of this research in neuroaids, which is also looking at these chaperone proteins.
Yeah.
Any other questions?
Okay, there's now a few
brain age algorithms that are used to, the same idea, comparing chronological age, brain age. Do you know, is there any work that's been done comparing the, brain age and the methylation age?
I don't, as far as I know, it hasn't been compared, but it will happen soon, I think. It's, I think a lot of people are interested in both markers, and it's an obvious, I think. Yeah. It's, if you have the data, you should do it fast.
Great. Anybody else?
But yeah, but it's a very, very interesting. Also, brain aging is an extremely interesting marker.
Good. Thank you very much.
Thank you for your attention.
Just, we do have a small tradition in psychiatry, so if you take the bother of trying to educate us, we also give you a small reward, a baseball hat that says UC San Diego School of Medicine, Psychiatry. Hopefully that will promote your bonding with our department. So,
there is no sun for now, but...
Well, you know, the rays still come through, so I would wear it if I were you.
Thank you, I really appreciate it.
Okay, thanks very much.
Thank you. Speak. Yeah.
Good chat.
Yeah.